Omicron's radical evolution. As nurses and doctors struggle with a record-breaking wave of Omicron cases, evolutionary biologists are engaged in a struggle of their own, figuring out how this world-dominating variant came to be. When the Omicron variant took off in southern Africa in November, scientists were taken aback by its genetic makeup. Whereas earlier variants had differed from the original Wuhan version of the coronavirus by a dozen or two mutations, Omicron had 53, a shockingly large jump in viral evolution. They found that 13 of those mutations were rarely, if ever, found in other coronaviruses, suggesting they should have been harmful to Omicron. Now the researchers are trying to figure out how Omicron defied the normal rules of evolution and used these mutations to become such a successful vector of disease. "There's a mystery here that someone has to figure out," said Darren Martin, a virologist at the University of Cape Town who worked on the new study. "Every time a virus replicates inside of a cell, there's a small chance that the cell will create a flawed copy of its genes. It could make the virus stick more tightly to cells, for example, or make it replicate faster." Over most of 2020, scientists found that different lineages of the coronavirus around the world gradually picked up a handful of mutations. In December 2020, British researchers were jolted to discover a new variant in England carrying 23 mutations not found in the original coronavirus isolated in Wuhan a year before. While some remained limited to certain countries or continents, the Delta variant, with 20 distinctive mutations, ousted Alpha and became dominant over the summer. And then came Omicron, with over twice as many mutations. Some mutations were shared by Omicron, Delta and other variants, suggesting that they had arisen several times, and that natural selection had favored them over and over again. But the scientists found a very different pattern when they looked at the spike protein that studs Omicron's surface and allows it to latch onto cells. Omicron's spike gene has 30 mutations. If a mutation were beneficial to the virus, or even neutral, scientists would expect it to show up more often in the samples. What makes these 13 mutations all the more intriguing is that they're not randomly sprinkled across Omicron's spike. 2 of the clusters change the spike near its tip, making it harder for human antibodies to stick to the virus and keep it out of cells. The 3rd cluster of mutations alters the spike closer to its base. This region, known as the fusion domain, swings into action once the tip of the spike has hooked onto a cell, enabling the virus to deliver its genes inside its new host. Rather than merging into the cell membrane, the whole virus gets swallowed up in a kind of cellular sinkhole, which pinches off to form a bubble inside the cell. Once the virus is captured inside the bubble, it can break open and release its genes. This new pathway to infection may help to explain why Omicron is less severe than Delta. The cells in the upper airway can readily swallow up Omicron in bubbles. But deep in the lungs, where COVID can cause life-threatening damage, coronaviruses have to fuse to cells, which Omicron doesn't do well. Scientists around the world have captured detailed images of the coronavirus. These 3 regions of the spike seem to have been important for Omicron's success. This makes it all the more puzzling that these 13 mutations were so vanishingly rare before Omicron. Omicron may have turned a batch of 13 bad mutations to its advantage by evolving under unusual conditions. People with chronic COVID infections can become evolutionary laboratories, hosting many generations of coronaviruses. Now it's stuck in this one individual, so all of a sudden it's doing things that it normally wouldn't do, said Sergey Pond, an evolutionary biologist at Temple University and an author of the new study. A mutation that allows a virus to evade antibodies isn't necessarily advantageous. It could make the virus's spike protein unstable so that it can't latch quickly onto a cell, for example. But inside someone with a weak immune system, viruses may be able to gain a new mutation that stabilizes the spike again. It certainly seems plausible, said Sarah Otto, an evolutionary biologist at the University of British Columbia who was not involved in the study. It's possible, for example, that the 13 spike mutations offer no benefit to Omicron at all. Instead, some of the other spike mutations could be making Omicron successful, and the 13 are just along for the ride. For example, it's not clear why, during a chronic infection, Omicron would have gained an advantage from its new bubble method for getting into cells. Subscribe to Fourlines Journal for more informational videos.